Good morning, everyone, and I hope everybody had a great 4th of July. We are enjoying this beautiful Friday morning here, and today we're going to dive into chapter 16 for our microbiology for health sciences textbook. And this chapter covers the second half of host defenses, as the first half was covered in chapter 15. Let's begin. First, we're going to talk about the major components of our immune system, and those include the lymphocytes, the markers, the MHC or the major histocompatible complex, and CD molecules, which are also known as cluster of differentiation. Lymphocytes they're central to the immune system, and um, they start out, they undergo development that begins in the embryonic yolk sac, and that shifts to the liver and also the bone marrow. And the in the final maturation of lymphocytes in B cells, they occur in specialized bone marrow sites, and in T cells that occurs in the thymus. Now, cells can express themselves each with several markers, and each marker has their own function, such as attachment to non-self for foreign antigens, binding to the cell surface that in cell surface markers that indicate self, such as MHC molecules, and receiving and transmitting chemical messages to coordinate the response and aiding in cellular development. MHC molecules are classified into three classes, which is class 1, 2, and 3. And class 1 MHC molecules, the genes code for markers that appear on nucleated cells. In class 2, the genes code for the immune regulatory markers. And in class 3, the genes encode for proteins involved with the complement system. And then lastly, the CD molecules, they are molecules that are involved in a variety of immune responses, and close to 400 CD molecules have been described. And B cell receptors, they pair up with the CD molecules and they bind to these antigens. Next, we're going to talk about immunologic diversity. And during the development of the T and the B cells, bone marrow and stem cells become granulocytes, monocytes, and lymphocytes. And the lymphocytes they will become either T or B cells, and the cells destined to become B cells stay in the bone marrow, while the T cells, they stay in the thymus. And then when the B and T cells reach the lymphoid tissue, each one is already equipped to become a single antigen. And inside this is the immunoglobulin, and certain lymphocytes will undergo a recombination process, and that codes them for the immunoglobulin. And the immunoglobulin is a place is the chemical class of proteins which antibodies belong, and they're large glycoprotein molecules that serve as the antigen receptors of B cells, and they're secreted as antibodies. Um, and they're the binding sites are highly variable, are are a highly variable in shape due to all the variable regions. And variable regions, or just abbreviated as V, are antigen binding fragments of an immunoglobulin molecule. And here to the right, you can see the stages of development of B and T cells that starts at the beginning in the bone marrow and then goes all the way down to a lymph node. Next, we're going to discuss the characteristics of antigens. all antigens, they are perceived as foreign, meaning that it is not normal for them to constitute in the body. And the materials for antigens fall into several categories, such as proteins and polypeptides, lipoproteins, glycoproteins, nucleoproteins, and polysaccharides. Um, Good antigens, they are chemical. Their characteristics are their chemical composition, their context, meaning what types of cytokines are present, and also their size. Lymphocytes, their capacity helps recognize and respond to only a portion of the antigens. So it is to discriminate the differences in molecular shape, and this is so their differences are so fine that it recognizes only portions of antigens, and the molecular fragments are called epitopes, and they're the primary signal that a molecule is formed. Right now, we're going to talk about activation of the T cells. Um, CD4 cells CD4 T cells are known as what is called T helper cells, and they bear antigen specific T cell receptors that bind to the antigens held by the MHC molecules. Um, and some of these cells are not fully differentiated or fully activated. Um, such and this includes things such as memory cells. And this cell population is activated and differentiated, but it stays uninvolved in the current response, which it will be ready to act in subsequent encounters with the same antigens very quickly and very strongly. Next is T cell independent antigens. Um, they are really simple molecules, and they um, they are usually such as carbohydrates with many invariable determining groups. So over here to the right, we can see in this little chart here from table 16.2 in our textbook, it's a chart of the characteristics of the subsets of T cell types with classic T cell response. If you want to pause right now, you can take a look and cover this quickly. Due to the short time period we have, we're not going to cover this in today's lecture. Next, we're going to talk about T cell response. And the response of T cells are mediated immunities and require involvement of T lymphocytes. Now, cell mediated immunities are a type of immune response brought about by T cells, and this is such as cytotoxic and helper effects. And in response to an HIV infection, T helper cells are greatly reduced, which is a major contributor to the pathology of AIDS. And regulatory T cells, they act to control the inflammatory process and to dampen the immunity and to make sure the immune response does not inappropriately target a human's normal biota. And cytotoxic T cells are active when they are activated. They recognize foreign peptides complexes, and they're complexed with self MHC ones, and these and that's presented to it, and they mount to a direct attack upon to the cells they are targeting. Lastly, we are to talk about on cell response is natural killer cells, or abbreviated NK. They are a type of lymphocyte, and they're related to T cells, but they lack specificity for antigens. They circulate through the spleen, blood, lungs, and probably the first killer cells to attack cancers and different types of virus infection cells. Next, we're going to talk about the immunoglobulins, and these are in different classes. And the immunoglobulins exist, and they're in structures, and they're also called isotypes. Now, the first class is a IgG, um, and it is a monomer with a Y shape, and it is produced by plasma cells, and its primary response in a primary response by memory cells, which gives it time to a given antigen process. The second class is an IgM, and these are very large monomers composed of 5 monomers, and they are also attached to the Fc portions of central J chains, and they have 10 binding sites included in them as well. The IdG monomers are found in the minuscule amounts of the serum, and it does not fit complement it does not fit complement aponocytes or cross the placenta, and its main function is as a receptor for antigens on B cells. The IgE is a monomer, but it is uncommon in blood complements and is usually, unless one is allergenic or has a parasitic worm infection. Its Fc region interacts with receptors on mast cells and also basophils as well. Next, we're going to talk about adaptive immunity. Now, adaptive immunity in humans and other animals, it is categorized as 2 sets of criteria, and that can they can be active or passive, and they can be natural or artificial. Active immunity is the immunity required through direct stimulation of the immune system, and it is categorized by 3 characteristics. It creates a memory that renders a person ready for a quick action upon re-exposure to the same antigens, requires several days to develop, and lasts for a relatively long time. Passive immunity, which is specific resistance that's acquired indirectly, and it is characterized by four characteristics, and that is the lack of memory for the origin, the lack of production of new antibodies, immediate protection, and short-term effectiveness. Now we got over immunity can either be natural or artificial. Now, natural immunity is any immunity that arises naturally within the organism through previous experiences with antigens, and artificial immunity is immunity that is induced by medical intervention, either by exposing an individual to an antigen or administering an immune substance. Now, lastly, we're going to talk about vaccinations. Now, active immunity can be conferred artificially by a vaccination, and a vaccination is a medicine that exposes a person to the antigenic components of a microbe without its pathogenic effects, so such as the flu vaccine, the COVID-19 vaccine, and so forth. And there are many factors that are considered when designing a vaccine, such as the antigen selection, how effective they'll be, the ease in administering the vaccine, how safe are these vaccines, and importantly, how much do these cost for the individuals receiving these vaccines. The preparation for for vaccines can be categorized either as whole organisms or part of organism preparation. And these categories also have 2 subcategories, such as are they whole cells, or are they viruses, or are they vaccines that only deliver antigenic components of the microbe in question. Now, over here to the right, this is table 16.5 from our textbook, and this is the checklist for the requirements of the effective vaccines. You can pause now and take a look at it, as we are not going to cover this in today's lecture. And that is all for today's lecture. I hope you all have a great weekend and a great rest of your week. And thank you all for taking the time to join me. And I hope you all have a good understanding of the material. Thank you.